Hello students, I'm Dr. Leena Meshram. I'm working as an assistant professor in zoology in Mahatma Phule ASC College. Today in this video lecture series, we'll talk about antigen. Antigens. Any foreign substance that enters into the body, that is foreign to us, that stimulates our immune response, it is called as antigen. So when an antigen, from the name itself we know antigen, it is antibody generator. That is any foreign thing which is not our self, which is not our body's molecule, it is something from outside. When it enters, when it is a non-self, then it is called as an antigen. So on introduction, when it enters into the body, it will generate an immune response. So any foreign substance that stimulates the immune system and produces an immune response, they are called as antigen. So these foreign things which generates an immune response in the body is called as antigen. So when you define it, a foreign substance when introduced into the human body, it can stimulate the formation of specific antibodies, or they are responsible to sensitize lymphocytes, that is B and T cells, and react with it. Okay, so the definition as such, it says that any foreign substance that stimulates the immune response, produces an immune response, it evokes an immune response, produces an antibody in the body, then it is called as an antigen. Now these are certain characteristics of an antigen, general characteristics of an antigen. An antigen should have molecular size more than 10,000, because the molecules which are less than 10,000 can bind with an antibody but cannot evoke an immune response. So generally the antigen should have high molecular weight. Secondly, chemically, when you see the nature of antigens, the antigens are proteins, they are polysaccharides. Generally, they are proteins, that is polypeptides, lipids, nucleic acid, and many of other material. They also functions as antibodies. So, biological nature of these antigens, these molecules are either proteins, they are either lipids, they are either lipoproteins, or they are nucleic acids. An antigen has an ability to stimulate an immune system, and then it is called as immunogenicity. See, when anything enters into your body and cannot induce an immune response, then you cannot call that as an antigen. For example, a stent, a rod which is in during an operation, a surgery, a stent, a pacemaker, they will not evoke any kind of immune response. They are inert, they will not evoke any immune response. So an antigen should have an ability to stimulate your immune system and to create and induce an immune response, and then it is called as immunogenicity. The antigen should have the ability to combine with an antibody. Unless and until an antigen combines with an antibody, the reaction will not start in the body, and hence it should have an ability to combine with the specific and the antibody. The property is called as antigenicity. So the very important properties of antigen is it should have immunogenicity, it should have antigenicity. When you say immunogenicity, it is the ability to produce antibodies in the body. When you say antigenicity, or it is also called as immunoreactivity, that is an antigen should react with an antibody. It should combine with an antibody. Antigens have specific structures that binds with the antibody, and those structures are called as epitope. Suppose Yes, we will see it in the next slide. Antigens will bind to the antibody at a specific site, and those sites are called as epitopes, or they are also called as antigenic determinants. So a single antigen, a single bacteria will have more than one determinants, or they will have more than one epitopes. Okay, so here this is a bacteria. This bacteria will have surface antigen. It can have certain DNA, or it is the nucleic acid which can behave as an antigen. It can have cilia, flagella, which can behave as a surface antigen. So a single bacteria may have more than one epitopes or antigenic determinant. So in this slide you can see this will behave as a different epitope, this will behave as a different epitope, this also will behave. So there are 4 different antibodies which will bind to this 4 different antigenic determinants, or the sites or the epitopes of the antigen. So what I said is a single antigen, a single bacteria, when you say it is an antigen, then it can binds to one, it can have one or more than one epitopes or antigen binding sites, which is also called as antigenic determinants. Now, types of antigens. When you say types, antigens are those molecules which are non-self. They are not your own molecules. They are something which is entering into our body. These are something which is induced into you, and then they will produce an immune response. So these antigens can be exogenous or they can be endogenous. So when we say exogenous, there are bacterial antigens, there are viral antigens, there are pathogens also, there are helminth parasites also, which can behave as which can be phagocytes, and which can be produced as an antigen. So bacterial antigen, these are antigen related to bacterial cell. Antigen which are secreted by bacterial cell, that they are exotoxins, or few of the enzymes which are released by the bacteria, which are again will behave as an antigen. So when these are outside the body, and when they enter into the body and behave as a pathogen, behave as an antigen, then we call it as an exogenous antigen. Endogenous antigen, in case of endogenous antigen, human tissue antigen, that is the blood group antigen, histocompatibility antigen, that is MHC protein, they can behave as an endogenous antigen. Okay, so blood group, we know that in case of ABO blood group, human blood group, ABO blood group, there are 3 different types of blood group, 4 different types of blood group: A blood group, AB blood group, B and O. We know that there is major compatibility issue, that a person who is A positive cannot take blood from B person. Okay, so why? Because every RBC cell will have a glycoprotein which behaves as an antigen. So blood group antigen, that is A blood group, will have A antigen, B blood group will have B antigen, and again one more antigen, that is Rh antigen, may be present. So this is again present in the body. We know that the blood groups will have these antigen. The blood groups are decided because of the presence of these antigen. Histocompatibility antigen, that is what I told you is MHC. And then again human leukocyte antigen, that is HLA antigen, may also come under endogenous antigen. So exogenous antigens which are coming from outside, endogenous and antigens are which are there in the body, and for others they will behave as an antigen. The chemical nature of antigen, as I told you before, antigens are proteins, they are polysaccharides, they are nucleic acids, and there are lipids. So basically antigens are biomolecules. They are biomolecules. Any inert thing cannot behave as an antigen, because they will not produce any
evoke any immune response. Some glycoproteins and phospholipids can also behave as an immunogenic, or they can also stimulate the T cells for cell mediated immune response. So chemically when we say, they are the chemical nature of the antigen, they are proteins, there are polysaccharides, there are nucleic acids, there are lipids. And in case of human blood group antigen, there are glycoprotein. I told you human leukocyte antigen, that is, there are glycoproteins. Glycolipid HB, again the blood antigen, P antigen is again RBC antigen. Proteins, they are proteinaceous nature. RH antigen is proteinaceous in nature. M and N antigen is again the chemical nature of the antigen is protein. Then we'll come to a next concept, that is, what is a complete antigen and incomplete antigen. Now complete antigen is an antigen which enters into the body. It will stimulate or it will evoke an immune response. It will stimulate your body's immune system and evokes an immune response. So when an antigen enters into the body, when it evokes an immune response, then we say it is a complete antigen. So a complete antigen can induce both immune response, and it can react with the immune cells of, or you can say it can react with the antibodies which are produced by the B cells. But when you talk about haptens, they are called as incomplete antigens. They are called as incomplete because they cannot initiate the immune response. They are not immunogenic. They can bind with the antibodies, but they will not initiate an immune response. So such antigens are called as haptens. Generally these haptens have molecular weight less than 10,000. So it acts as antigen only if it binds to a carrier protein. So in case of these haptens, they are bound with certain proteins which are called as carrier proteins, and then these antigens, this is a native carrier protein and these are the haptens or antigenic determinants which can bind with a carrier protein, and then it can be introduced as an antigen. So they are native determinants and haptogenic determinants. Haptens have generally low molecular weight substances. These substances are not immunogenic by itself. As I told you, when they are bound with the carrier protein, then only they will behave as a complete antigen. If coupled to larger molecules like albumin and globulin, they can behave as a complete antigen. Certain examples of haptens is penicillin, sulfonamide, aspirin. There are certain cosmetics, tranquilizers, neomycin, skin ointment. These are the examples of incomplete antigen. Thank you so much. I hope you understood the concept of antigen.